Hello friends, welcome to BMH Learning. This video deals with clone by clone sequencing. Clone by clone sequencing is a method of genome sequencing. It requires the mapping of each chromosome prior to DNA splitting. After mapping, the DNA should be broken down into fragments of 150 kilobases long. These fragments are then ready for sequencing. Next step is the insertion of DNA fragments into bacterial artificial chromosomes and then put inside bacterial cells to grow. The chunks of DNA are copied each time the bacteria divide to produce lots of identical copies. The DNA in the individual bacterial clones is then broken down into even smaller overlapping fragments. Each fragment is 500 base pairs long, so that they are a more manageable size for sequencing. These fragments are then put into a vector that has a known DNA sequence. The DNA fragments are then sequenced starting with the known sequence of the vector and extending out into the unknown sequence of the DNA. After finishing the sequencing, it is necessary to identify the areas of overlapping sequences. Thereafter, joining of the fragments takes place to form the larger fragments initially presented in bags. Next, the assembly of larger fragments into the chromosome takes place according to the genome map. This was all. Thanks for watching.